So, welcome back to the toxicology class again. After a break, we are again with a new chapter, that is snake bite management. Myself is Professor Shomnath Dash from the Department of Forensic Medicine and Toxicology, RG Kar Medical College, Kolkata. So, we begin with the anatomy of a snake. If we just look at the picture of this snake, we'll find that the snakes are not having any kind of limbs. What it is having is a long slender body, as you can see here, and it ends at a tail. The entire body is covered with the skeleton system, but the most important part of this snake is the head part, which is having the medically significant structures like that of the fangs, the venom duct, or that of the tongue, as you see here. If we just look at the curvature of the venom duct, we'll see that the curvature of the venom duct is inward. Both the curvature of the fang as well as the venom duct is inward, as we see here. Now, the most important structure, that is the fang, if we look it in a detailed way, we'll see that it is having a venom canal inside it, looking just like that of a hypodermic needle. So whenever the snake is putting its fang on its prey, just like that of a hypodermic needle, the venom is channelized through this venom canal and is getting inside into the body of the prey. How it is being channelized is shown in this picture. This gland is known as a modified parotid gland of the snake, also known as the venom gland. Whenever these compressor muscles compress during the prey time, this gland is squeezed and the venom travels through this primary venom duct, then through the secondary venom duct into the fang, and then into the body of the prey. And here is a real-time picture of the snake where the venomous gland is being pressed by the two fingers, and you can see that the venom is coming down the fangs and it is being collected. Now we come to the classification of the snake. The four major group of snakes are classified as the Colubridae group, the Elapidae group, the Viperidae group, and the Hydrophytidae group. As far as India is concerned, as well as the entire world, the two most important groups are the Elapidae group and the Viperidae group. And these two groups are highly significant because they causes the major case fatalities throughout India as well as the major part of the world. The Elapidae group includes the cobras, the kraits, the mambas, and the coral snake. The Viperidae group includes the pit viper, the Russell's viper, the saw-scaled vipers, etc. We are not very much concerned about this Hydrophytidae group because they include the sea snakes. Okay. But the major Colubridae group includes the mostly non-venomous snakes, which has a very large classifications and the examples. So we are not going to discuss in details here. Another important question which is often asked to you people are the difference between a venomous and a non-venomous snake. This group is the venomous snake group and this one is the non-venomous snake group. If we just look at the picture, then we can have at least a few difference between this venomous and the non-venomous. Though the difference between them as written in your textbook of forensic medicine and toxicology are a whole lot, but the major differences has been described here. Just look at this pupil. As far as the venomous one, it has an elliptical pupil, while this is a round pupil as far as the non-venomous group. The most important difference is definitely the presence of fangs, which is present in the venomous, and in a whole lot of non-venomous they don't have this fang. And the shape of the head, you see it here, the venomous snakes are triangular, head is triangular, while the non-venomous it is more or less rounded in characteristics. There are difference between the size of the tail, the abruptness of the tail. There is a difference between the scale that is often being found over the belly. But as far as these picture is concerned, there is another very important finding that I must tell you, is that the difference between a venomous and a non-venomous snake can also be made by the use of the labial scales. What I mean by this is that these are actually the scales that is surrounding the mouth part. If we just look at this venomous snake, we'll find that the third group of labial scale, it actually touches the eye or beyond that. But as far as the non-venomous snake, you will find that these labial scales, that is the scales that is surrounding the mouth, they directly touches the eye as far as the first layer is concerned. So this is a very significant differences. So you people can remember a whole lot of differences that is there in the book, or a few that I have already stated in this picture. Now let us look at the pictures of different snake which are medically important as far as West Bengal or even the entire India is concerned. So let us learn something about the snakes. This first snake is known as the Russell's viper. In Bengali we call it the Chandrabora. It is a highly venomous snake. The head, as you can see it here, is triangular in shape. It is broad, and the entire body is also broad. And it is not having any kind of hood, but the major specific identification is the diamond shaped brown or circular marks that is present over the entire body of this Russell's viper. They are very dangerous, and most of the victims, they require hospitalization if they are being bitten up by this Russell's viper or Chandra. The next slide shows the saw-scaled viper. The snake can be identified by the presence of the saw-like scale over the entire body, but from a distance it can be identified by the presence of rhomboid shaped structures all over the body, as you can see in the picture. And here is a real-time picture of the snake where the venomous gland is being pressed by the two fingers, and you can see that the venom is coming down the fangs and it is being collected. This is called the Cobra. In Bengali we call it a keute. The color is usually from dark gray to black. In some of the cases we can also have other kind of cobra like this one. It is white or yellow is in color. Sometimes there are bands present like that of a white band and the yellow band present all over the body. The most characteristic sign is the presence of a hood, and that hood is also having a lotus sign on the posterior aspect. They usually come out during the night and feed on the frog, rat and the small birds. There is another snake we call it an Indian cobra. It is more or less like that we see here. We call it a gokro in Bengali, and it is brown in color wood is having a spectacle-like mark. So the difference between a cobra and an Indian cobra is that the spectacle sign and the lotus sign that we have discussed just now. Now let us come to the discussion on venom. What is this venom? The term venom derives from the Latin word venuum, which means poison. This venom system is an important adaptation that has evolved in the different animals for certain reasons. In most of the countries, the species that we have seen, venom is actually a conglomeration of different kind of enzymes. And with the evolution, we have seen that these enzymes are being added one after the other in order to make the species much more dangerous to other animals on which they prey. Snakes are usually found in every continent except in the Arctic and the Antarctic region. There are a few other countries where we do not get snake. Just look at this picture. As we have already told, that initially the snakes were non-venomous, but with the time of evolution, the snakes have adapted to include a lot number of venom in it. This is we call the early venomous snake, which is having one or two type of venoms. But as the evolution progressed, we find that newer species are being created. They are all having different kind of venoms or the combination of different kind of venoms. So this type of species that is now present are much more dangerous than that were present earlier. If we just look at the chemistry of the snake venom, we will see that mostly they are acidic in nature and soluble in water. As I have already told, that the composition of the snake venom is mostly the enzymes. And there is a long list of enzymes that we will discuss very soon. If we classify venom, we have four types of venoms. The first one we call is a neurotoxic, the second one the cytotoxic, the third one the hemotoxic, and the fourth one is myotoxic. As far as Indian snakes are concerned, we are mostly interested about the first three groups, the neurotoxic, the cytotoxic, and the hemotoxic. To be more precise, the hemotoxic and the neurotoxic. We are not very much accustomed with snakes which are having the myotoxin. Mostly these are hydrophytes, that means the sea snakes, which are very rare as far as India is concerned. This is something about the neurotoxic venom. As the name suggests, this type of venom usually attracts the nervous system and are mainly of the fasciculins, the enterotoxins, alpha neurotoxins are the enzymes that are usually found in the neurotoxic venom. Now these fasciculins attract the cholinergic neurons by destroying the acetylcholinesterase. So the acetylcholinesterase enzyme is not there. Naturally, the acetylcholine cannot be broken down and it stays in the receptor end for a long time. I think you have gone through all these names, which is known as the tubocurin, the cobrotoxin, and the bungarotoxin. All these block the acetylcholine receptor or prevent the opening of the ion channel. Now, by blocking these signals from the nerve to the muscle, these toxins causes paralysis, and this paralysis can result ultimately into death if it happens to be mostly over the respiratory muscles. It takes around 10 minutes for the venom to affect the nervous system. So most neurotoxins are too large to cross the blood-brain barrier. Naturally, they do not enter into the brain. But the spinal cord is easily being damaged, as well as the local action causes the most of the case fatalities. So welcome back to the toxicology class again. After a break, we are again with a new chapter, that is snake bite management. Myself is Professor Somnath Dash from the Department of Forensic Medicine and Toxicology, RG Kar Medical College, Kolkata. The next slide shows the saw-scaled viper. The snake can be identified by the presence of the saw-like scale over the entire body. But from a distance, it can be identified by the presence of rhomboid shaped structures all over the body, as you can see in the picture. This is the picture where we'll see that how these phospholipid molecules are being separated by the cytotoxins. This one we call this cytotoxin, the cytolytic toxin, you can also say. This is the toxin here, and this the protein lipid protein layer, as we see here in the cell membrane, are being lysed by this cytotoxin. And when this thing happens, that means there is a rent into this lipid layer and the phospholipid layer, to be more precise. Naturally, whatever there is inside the cell, they comes out, especially if it happens in the endothelial cell membrane. Then the blood which is inside the cell, that will come out and will result in the formation of hematomas in that specific area. The last of these type of venom is the hemotoxic venoms. This hemotoxic venoms usually causes hemolysis or induces the blood coagulation, as it is written here. If we just remember our old days study, then we can know that the process of coagulation or the blood coagulation usually occurs in two pathways. One is called the classical pathway, and the other one is known as the alternate pathway. And the causative agents of these alternate pathway include a whole lot of things, of which snake venom is another one. So here the snake venom, which triggers up the blood coagulation inside the blood vessels, and when this occurs, we are very commonly encountering a process which is known as disseminated intravascular coagulation, or the DIC. Naturally, the whole lot of cardiovascular system, the circulatory system, and the muscle tissues, they are being destroyed because of this disseminated intravascular coagulation. This is the main mechanism by which the vasculotoxic snake bites causes death. vasculotoxic snake bite, as we have seen, is mainly done by the snakes which are vipers. The next three slides we will show the different type of enzymes that is present. We have already discussed the broad classifications of these kind of enzymes. You people have to just remember this chart, because sometimes in your MCQ questions we get these kind of answers or MCQs where you can have to identify what kind of toxin and what is its action and where it is commonly present. This is the first slide. This one is the second slide. You can remember the names of these enzymes, and whenever required you can give the answer to those questions. This one is the last slide of the three slides, which contains the names of the enzymes and where it is present and what are their actions. You can remember it, as I have already told, for the MCQ purpose. In this context, I would ask you to download an app which is known as the Snake Bite and Poisoning app, present in the Google Play Store. And in that app you will get a whole lot of information about different type of snakes and the first aid for the common people. The snakes are being depicted by picture, and all the information regarding the snakes are given in that app. It has been prepared by the Poison Information Center. It is also known as Obilombe. You can search it by giving the name as Snake Bite and Poison Information app in the Google Play Store. So this much is for today's discussion. In the next class we will go into the detailed discussion about how we will manage the cases of snake bite. Till then, goodbye.